We're running a differential expression analysis, end-to-end. No R needed. BioMate matches your request to a curated bioconductor workflow and drives every step. First, it embeds your request against the bioconductor knowledge base. The panel stays empty. No pipeline is chosen until the match resolves. Two curated chains score close, a count matrix DESeq2 path and a fuller FastQ pipeline. BioMate shows both with scores and pre-selects the top one. With the DESeq2 chain committed, the panel fills with 6 phases: QC, normalize, DE test, shrink, enrich, visualize. Why DESeq2? A raw integer count matrix needs a negative binomial model, not lim-of-oom, which is for continuous intensities, routed by data format, not habit. Execution starts. The active phase auto-expands. Edgers filter by expr drops genes under 10 counts. Parameters and outputs appear inline as each step finishes. Every step opens with the workflow's defaults already filled in. Click the gear to adjust a value inline. Change only what you need. Each phase has an editable QC gate, pre-filled with its default threshold. The auto loop switch lets a failed gate fix its own params and rerun. When auto loop is on, a failed gate posts a halt box, the failing metric, the proposed fix, and a 10-second countdown. Apply, pause, or dismiss. As each step finishes, its files drop into the panel. View opens them in line. Download grabs the file. No waiting for the full chain. Any running phase can be stopped. Finished steps and outputs are kept, then resumed exactly where you left off. After normalization, counts, metadata, and size factors fold into one DESeq dataset, the typed object every downstream step reads from. Phase C runs the walled test on design tilde condition, 2,841 genes at 5% FDR, producing a results table of fold change and adjusted P per gene. Before ranking, AppRGM shrinks noisy low-count fold changes towards 0, so enrichment isn't dominated by extremes. clusterProfiler's GSE-GO runs on the ranked list, top term, regulation of apoptotic process. Phase F renders the volcano, dot plot, and heat map. All 6 phases done. DESeq2, APEGLM, clusterProfiler, enhanced volcano. Chat drops a findings card. The panel flips to the after-run report. The report has 7 tabs in reading order. Results first, methods and reproducibility next, audit last. The whole thing exports to one document. Findings: the headline metric, a few bullets, and figures read straight from the manifest. Nothing is invented. Uncited claims are tagged author judgment. Outputs lists every file by phase, the results, object, volcano, go, dot plot, top 50 heat map. View previews inline. Download grabs one or all. Download isn't the end. Every results object has an Open in RStudio button, a live R 4.3 session with DESeq2 loaded, and your dataset pre-mounted. Clicking View on the volcano renders it inline. Fold change versus significance. Up genes red, down blue. BioMate picks the viewer from the file type. Methods is paragraph form prose, tools, versions, exact parameters, written so a peer can replicate the run. Paste it straight into a manuscript. Parameters lists phase, step, and value for everything the run touched. A ready supplementary table. Defaults, overrides, and auto-loop tweaks are distinguished. Software pins every package version: DESeq2, APEGLM, EdgeR, clusterProfiler. With parameters, it's the reproducibility baseline. Citations auto-generates the reference list: Love 2014, Ju 2019, Wu 2021, and more. Methods and citations export together. QC audit logs every gate that fired: gene retention, size factor range, replicate count. with threshold, observed value, and action. 21 CFR, Part 11, friendly. That's the full DESeq2 thread. Match, route, run 6 phases, a 7-tab report, all from one request. Download wraps it into a single document.